That was pretty good. It wasn't Run the Jewels, but it was pretty good. Thanks for having me again. I'm glad to be a perennial favorite. So my name is Will Salerno. I'm the Director of Genome Informatics at the Human Genome Sequencing Center at Baylor College of Medicine. We're one of 4 large-scale sequencing centers funded by the NIH. So today I'm going to give you the perspective of what's going on right now in terms of what we call discovery commons or data commons. We sequence a lot of data. What do we do with that? And of course, a lot of that involves working with DNAnexus. Oh, it works. Is one of these a laser pointer? There we go. So, Human Genome Sequencing Center. Here is a picture. Lots of people are asking, and I genuinely do appreciate everyone asks if I was okay after we got rained on for 30 years. I rode my bike down into the medical center. That's how I would normally drive into work. That's about 50% floodwater and 50% sewage. So it was not pleasant, and I have to buy a new bike. But lots of people got very lucky. Lots of people are struggling. If there's anything you can do to help, please continue to help. It did get bad down there, but I was lucky. Human Genome Sequencing Center, we did not miss a beat. We did not slow down in our sample throughput at all. We were prepared for this because we've been at it for 20 years. We have 180 well-trained staff, and we have 30 sequencers that we kept running throughout the entire storm and all of the debacle after that. Here I have just some of the partners that we work with, some of the organizations we sequence. We're at Baylor College of Medicine, but we really are our own entity, and we partner heavily with UT Health, with Dr. Eric Borwinkle and his group to do a lot of these large studies, which range from cardiovascular disease to a lot of clinical work, which I will get into. But today I really want to focus on the work that we do with the NIH and their sequencing programs, which NIH has 27 institutes and centers. I'm just going to talk about stuff we do with 3 of them, NHLBI, the NHGRI, and the NIA. This is the alphabet soup slide, so you know what I'm talking about moving forward. NHLBI is a lot of the top med program. This is cardiovascular disease largely. They have about 130,000 whole genomes that they've sequenced with us and with other sequencing centers involved in top med. And they're also shifting into the multi-omics space, RNA-seq, metabolomics, et cetera. And it's sort of similar to the CCDG, the Center for Common Disease Genomics. There's a bit more broad of selection of diseases, including autism and bone disease, as well as cardiovascular. This is what was evolved from the old large-scale sequencing and analysis centers, which is the LSAC. Lots of different cohorts. I put charge down here because, if you've been to one of these Nexus Connects before, or you've heard Nexus talk about some of their flagship projects with the HGSC, they talk about doing the charge commons. Charge is a collection of cohorts focusing on aging and heart disease, and that group gets their samples sequenced through TopMed and CCDG and other programs. So a lot of times these terms can get used interchangeably, but this is sort of the structure. NHGRI also has the eMERGE network, which is sort of the other end of the spectrum. They're doing a custom clinical panel with about 9 clinical sites. I'll get into that. But one thing to point out is that we have been driving that sequencing along with the other sequencing center at our newly deployed, open, validated HGSC CL, our internal clinical lab, which did just open this year, which is a big accomplishment for us. And lastly, the Alzheimer's disease sequencing project, which has been going on forever. That's a bit smaller data, but there's a lot of rich phenotype there, especially with the family data. So when I'm going through the data sets as examples today, these will be the data sets I'm speaking of. What are we doing with these data? We're not just a contract sequencer where you give us your samples and we sequence them. We have a vision. We're at a College of Medicine, and this is the virtuous cycle that has gotten a lot of play over the last couple years. And the idea is that there's gene discovery, which goes into translation, which is bedside, which feeds data, which feeds the discovery. And this is sort of the idea of the cycle that we're working on. These are some of the specific mechanisms. And then these are the supporting resources for these activities. And really talking about the data commons, which is what I'm talking about today, capacity is informatics, is how do we not just share the data. How do we get the data and really extract useful information from the data? So I would say for data commons, there's 3 things that I focus on. Clean the data are messy. Have flexible methods. You don't want to be a one crank machine, right? Lots of places do this. You build a machine, you have one crank you pull, everything goes through that way. That's really well suited for a lot of operations, okay? But when we're at the NIH, we have multiple partners who need multiple different things. We need flexible methods. And the whole point of a discovery commons or a data commons is to bring people working on different things together. So flexible methods is probably one of the big differentiators between us and something that's a more high throughput, focused in one direction machine. And the last thing is interactive tools, okay? Having the ability for people to observe the data, do their synthesis, and put the data back into something that can inform other people's analysis. So that's our vision of what we do with these data and my vision for how we implement that in the data commons. Usually when I give a talk, especially to a room full of people who are interested in informatics, a lot of it's about scale. I would have one slide about scale, and this is what we do at scale. It's a job a minute, it's a quarter of a terabyte an hour, it's 2 terabytes a day, 1 million CPU hours a month, 9 petabytes a year, and 2 petabytes perpetual data live. That for various units of what I just said. This is what we have to support at scale to do about 2,600 whole genomes a month. So my primary job is to make sure that doesn't break. So when I have free time, when that's done, we work on all of this stuff. That compute, all of those requirements, is just our normal whole genome throughput. It doesn't include all of this other work that we do at a research academic institute, like the HGSC. What I want to focus on today is this stuff, not how we do the sequencing at scale. I talked about that last year. DNAnexus has done a great job facilitating a lot of other places doing similar type at-scale sequencing. What do we do once we have the machine running and it's flexible to support heterogeneous data? So here's a run-through of some of the analysis, independent of our production throughput with DNAnexus. Here's a summary of some of the analysis we've done on the cloud. Our charge, again, was one of our big flagship projects in the beginning. We published a great paper working mostly with Andrew Carroll on structural variation using long reads. More charge, we did ADSP, we did eMERGE portal. The things I'm going to focus on today are some of the reprocessing we did and the challenges involved in that, how we did a 24,000 whole genome call set with relative ease, and some of the software we developed, and then talk more about eMERGE, our partnership with Sentient via DNAnexus, and then charge-free 6. And honestly, I would have been happy to get up here and give a 3-minute presentation on one slide, and that'll be at the end, because I'm so excited about what we're doing. But I'll go through this other stuff. It's not that bad, I promise. I'm going to start with the NIH Whole Genome Cloud pilot, okay? The idea was we have 24,000 whole genomes sequenced at 4 different sequencing centers in 4 different protocols. How do we combine those in such a way that when we hand the data over for people to use, it can be used without excessive amount of QC or cleaning? So the idea was, let's clean this up to begin with. So to do that, we spent literally a year coming up with a protocol of going from a fast queue to a cram. A year figuring out how to do that. So imagine you have like the 4 nerdiest bioinformaticians ever sitting in a room arguing for a year about what kind of quality score binning and what logarithmic scale to use in order to get that right. A year. I had 2 people quit because of this project. But what you can see is that it worked out well because we took samples, the same samples, 15 of them, processed on each of the original pipelines and looked at something like chimeric rate, which is going to heavily influence your ability to accurately detect structural variants, which is one of the reasons you do whole genome sequencing. Wildly different. This type of noise, structural variants are hard enough, we don't want to introduce it. After we standardized the protocol, much, much more even. And the slight differences that you see are something that we can explain and characterize and deal with. This is a very heterogeneous data set. A wide mix of coverages. It's a huge data set, about a little more than half a petabyte. But we went through all of this, and now we have a hardened protocol. That was something that we worked on with DNAnexus, but also I want to just point out that Sention worked with us on this, and you might know Sention because they made everything really fast. Fast is great. What they really did that is so valuable to the harmonization process is they did it exactly. I don't care that they did it fast, because if it's slow, it just means it costs me a bit more money and I can find money. What I need is something that is exact, so when I swap it in, I don't have to explain it to anyone. Okay? So this idea of precision and being exact is a really important when we're operating with heterogeneous data at scale. So when we have this protocol, now we had to do the processing. Here are some of the stats, millions of CPU hours. We ended up generating like a ton of cram files in 6 days. I was ready to kick all of the jobs off, and I'm like, you know what, let me email Andrew Carroll real quick and make sure I'm not gonna break anything. I'm like, hey, I'm about to kick off like a whole bunch of 32 core node jobs. And he's like, please don't do anything. Let me do this for you. You will break AWS. So I didn't break AWS, that was a good thing. A lot of work went into building our Parliament structural variant and variant calling and align stats app, which I'll go into in a minute. And then here are just some of the numbers of the efficiency improvements we saw by folding Sention in. But again, the efficiency wasn't important. Sention and Nexus worked out the way to do duplicate marking on supplemental alignments that had to be super exact, and if you tried to cut any corners, it was wrong. These were very in-the-weeds problems which needed to be perfect, and we figured those out. It took time, but we figured them out, and we got it really cheap. So to the Sention guys where you are, thank you very much in public. We do appreciate that. The summary of all of this is that it turned out to be just under 23,000 samples by 4 of the CCDG centers. Here's the breakdown. This is the total number of bases. I can't count that high, but it's a lot. Overwhelmingly high quality, low contamination. Median coverage, 36x, although like I said, there's a wide variation there. These data are not publicly available. Okay, so I think the point that Jeff made earlier about one of the big resources of the day that he's working on becoming public, that's fantastic. One of the big challenges we have when you're dealing with heterogeneous data is your consents are also heterogeneous, okay? So it's really hard to get these data into everyone's hands. I don't have a solution to this. I'm just saying you can't look at this right now, and I wish you could. When we're assessing the quality, though, to get back to the point about cleaning the data, this is a chart of average coverage versus the percent of bases that have at least 20x. So you see this sort of expected shape where as your average coverage goes up, you saturate the number of bases that are covered at 20x, and the bifurcation is just gender that you see. The different colors are different sequencing centers, which I've hidden so no one gets embarrassed. We did really good, though. But you will see that there's this smattering of things that sit under the curve, where even when you have high coverage, you're not getting smooth, consistent coverage of the bases at sufficient coverage to accurately call a variant. This is what worries me. This is the stuff that should keep you up at night if you're sequencing at scale, because relying on one metric like average coverage isn't sufficient. You have to look at many, and you have to look at them in a population context. So speaking of which, that was just some basic metrics on our basically our alignment statistics, but we also have done a ton of structural variant calling. And this is work that was started by Adam English at the center a long time ago, and it was sort of picked up by Andrew Carroll, who went crazy and ran with it. And now we have Fritz Sedlacek in the group as well, who's working on it. We have this new Parliament 2 app on Nexus. It's multiple structural variant callers that are optimized for the cloud. It merges those calls, and it does orthogonal genotyping with an orthogonal method. For this project, we added in some additional features, including our internal align stats, which generates 150 metrics from the CRAM, which is really useful when you're trying to assess quality. We get contamination assessments. We have Sention in there to help with the realignment, our small indel caller, and it runs $5 to $8 a sample, okay? And it is as efficient as you can get it. The 5 to 8 variation is just your coverage, right? Because you can have a doubling of coverage in these samples. So for this 22,000 set, we ran 5 of the callers. We got 6.2 million structural variants after merging. And as Andrew pointed out, there's over 1 million of them with at least 1 in 1,000 allele frequency and has an orthogonal genotype. I think I said that right, Andrew? Good. I just copy what smarter people say. That's my job. And what we can see here is the allele frequency spectrum, which does recapitulate what we'd expect sort of based on SNVs and indels. So we're on the cusp. If I had another week, I'd have more data to show you, but this is a huge resource of structural variants on large whole genomes. And this is something, even if we can't make the individual calls available, Andrew and I are committed to making at least sort of the background allele frequency spectrum available to individuals, because I think this would be a great annotation resource for people who are looking for what does population structural variation look like across something that's pretty diverse. However, going back to the assessing quality, you can't just make the calls and be done with it. I showed a variation of this slide last year, but I think it holds up. This is ADSP data. It's 584 samples, which includes 3 replicates, 3 samples that were sequenced 3 times, once at each under 3 different protocols, okay? 3 different sequencing centers. You do a PCA of these structural variants, they separate by center. Okay? Not only do they separate by center, the replicates, this triangle is the same person. So the divergence of structural variants, if you don't clean them, is so strong that the same individual separates based on where it was sequenced as opposed to who it is. That's how noisy structural variants are. If you isolate the protocol-specific ones, structural variants that are unique to a sequencing protocol, you pretty much get your 3-dimensional axes, right? So there are a lot of these and you have to deal with them. However, if you just, and I don't have this slide up there, you just have to trust me because they all look the same. If you take those protocol specific or those protocol unique sites out, you still get separation by center. The replicates, you still do not observe replicate collapse. You really have to go and not just say, okay, give me all the structural variants that are unique to one center, but you have to be fuzzy about it and say, okay, it's mostly preferentially in one center, but it can occur at another one. One of the guys in my group, Waleed Nasser, did some really great work where he came up with some rules to extract structural variants based on their preferential ability to, or presence with different centers, and we were able to get the replicate collapse that we were looking for. And this replicate collapse is what really drives a lot of our QC. Mendelian consistency and replicate collapse. You should not be seeing impossible situations. If it's the same individual, it should behave the same way no matter where it goes. And the great thing is once you QC these 584 samples are actually they're sparsely related across 111 families. From just those harmonized structural variants, you can recapitulate the family structure. Maybe that's surprising, maybe that's not, but I don't know how often something like this has been done with whole genome structural variants. So that was my pitch for whole genome. You have to be careful, you have to be accurate, and you have to clean the data. And we figured a lot of that out, and all of these tools and methods, they're available on DNAnexus, and we're happy to work with other people who want to figure these out, because the more problems you bring to me and that we're able to fix, the more I know how to fix my next data set. That's part of the cycle. The next thing I want to talk about quickly is the eMERGE program, which is the other end of the spectrum. This is doing clinical sequencing. It's 9 clinical sites, 2 sequencing centers. There's a panel of about 100 genes and 1,500 small variants. And the idea is to reconcile these with clinically actionable phenotypes and medical records, and do all of this in a CAP-CLIA environment. So this is just a map of where everything happens. So we have our normal pipeline for processing and generating the VCFs, and then we have Neptune, which is our automated clinical reporting. The heart of Neptune, and I think I went through this last year, but the heart of it is a VIP data set, which is here are all the variants that we think are clinically actionable. And what it does is it filters your VCFs through that, gives out a negative pre-report if it finds nothing, gives out a known positive report if it finds something. If there's a novel positive, which we have through our codified work, where we have a way of sort of predicting, hey, we think this might be relevant, it cycles back into the VIP. Bottom line, the more that you review, the more that your VIP grows and the more that you're able to automate. That's the idea here, is that you're interactively capturing knowledge of your clinical sign-out. And this Neptune tool is something we have locally and that we're actively working with Nexus to deploy in a way that can be shared. This is a summary of our eMERGE results, what kind of variants we found. It's not a lot of positives. It's doing a lot of negative reports and getting them out there. But for those positives, because there's 2 clinical sites, and this is an NIH collaboration, we get a lot of feedback on whether 2 people reviewing the same thing come to the same conclusion. It's surprisingly not that common that they come to the same conclusion, even when they're looking at the same data sitting next to each other. Clinical sign-out is hard. Putting it in a commons, automating as much of it as possible is sort of critical to being able to do this at any high throughput. This is only 15,000 samples. I mean, I see numbers in the hundreds of thousands that are being thrown out that we would want to do in some sort of clinical setting. Automating this is going to be absolutely critical to getting the information extracted from the data. Part of what we've built toward this are our eMERGE portals. We've built a couple at DNAnexus, and we've built a couple at the HGSC, and I just want to give some feedback because I'm going to immediately leave this meeting and go get yelled at for an hour for not having this done already. So I want to get some credit for it. This is what we've built at the HGSC. It is a data warehouse. If it weren't, I don't know how to make it not a data warehouse. I'm not even sure what a data warehouse is. I think we just made that up and then found out it was real. But what we do have is all the variant data loaded with the phenotype data and annotation based on our predictive codified software. And this is something that's searchable. This is a discovery tool. It allows individuals to unify the genotype, the phenotype, and clinical annotation and grow that VIP database in order to, find subsets of the data that might be linked in some way that's actionable. This is something that we've built locally because we had certain resources and partnerships that allowed us to get this done with the idea that we then expanded out once this pilot was complete. In parallel, working with the DNAnexus team, specifically Martin, we've built the eMERGE Cloud Dashboard, which is this very slick data management tool where for all of the samples in eMERGE, which is an ongoing process, clinical sites are submitting their data, we're sequencing them, we're processing them, we're reviewing them. They're asking every 3 minutes, where's my data, where's my data, where's my data? Just go check the website now and leave me alone. That's basically what the title of this should be. Stop asking. But this allows you to filter the data by all of the different fields that we load. And we don't really want to encourage people to download their raw data. We want them to do the work on the cloud. That's the whole point. But we do want people to be able to process in a way where if they filter their set, they're able to operate on that. So we have the functionality where you operate on the set, you get the subset that you like, you select them, and then we give you the records, right? Because right now at this stage, this isn't a really polished tool. What this is a development platform. You're able to filter your samples to the ones that you want, and then get a set of objects or IDs that allow you to build around them. These record IDs contain all of this meta information. So the idea is we now have a mechanism where anyone can develop apps where we've done the filtering part for you. You can go and pick your set. Now, if you want to build something that takes your subset and executes an analysis on Nexus, you build that app, you take that record list I showed you as its substrate, put it in. This is how the development process goes. We build the apps, and once they become hardened and we want to put them out, we work with the DNAnexus engineering team to make that a blue button where you just press it and it happens. So this is part of the cycle of, we're experimenting. We don't exactly know everything we want to do, but we facilitated the ability to figure that out. Here's my favorite slide. I don't know how much time I have left. 5, Jesus, I'm talking fast. I will go slow and enjoy this because I've been waiting for this for years. I came to the Human Genome Sequencing Center off the street. Jeff Reed hired me because all of the people he wanted to hire had visa issues. So he's like, I need someone who's literally doing nothing with their life but can come and press a button for me. And I'm like, hello. And one of the first things when I showed up that Jeff and Narayan, who joined shortly after, worked on was making a project level VCF. The idea that you have your sample level VCFs and you want to create something that, you put them all together, but there's all these, it's a sparse matrix that you have to fill in. And Jeff and Narayan came up with what at the time was a really clever way of doing that within certain constraints we had at the center. And Andrew Carroll helped execute that. And we did these charge PVCFs, which were, 11,000 exomes, about 4,000 genomes, 3.3 million compute hours, all of these concurrent cores, a petabyte of data. And then the next year we did an even bigger one. It was more data and it was amazing. And they gave us an award for it and it sits on the top of a shelf somewhere. Got an award for doing what at the time was the largest compute ever. And I always like to make fun of it because it makes me laugh. But really, it's not about being the biggest compute. This was a huge exercise in logistics and coordination. And it was truly impressive. And the award is well deserved. But what's even more impressive to me now is we have now switched to doing GVCFs, which we didn't have before. It's our new Atlas version. We have the GVCFs. But we didn't have a way of merging them at scale. But DNAnexus did. And I got an email, and this is something I've been telling my bosses and collaborators. I'm like, I will deliver this. I will get this to you. And we got the GVCFs early this year. Jesse on my team did a great job with that code. And yesterday, Mike Lynn at Nexus emails me. It's like, it's done. And the merge took 24 hours and cost $300 or something. And I just started emailing everyone. It's like, it's finally done. What's impressive here is that we had to do all of this work and spend, do the largest compute ever to do 15,000. 2 years later to do 621 more. What's really like, oh, do we really want to spend a bunch of money to redo this giant thing? Now it doesn't matter. Because of the innovation, I can do this every single day if I want and not break it out. I can do 15,621 today. Someone's like, oh, you forgot to put a sample in. Before, oh, it'd be a huge ordeal. I'd have to go fix everything. Now I'll just go run it again. Okay? This is the innovation. It's not about doing these big computes at scale. It's about making the hard things easy and then automating the easy things. And that's exactly what Mike did. So again, thank you for that. The fact that, I don't even know how many concurrent cores. It doesn't matter. It just got done, right? That's the thing, is that a hard thing became easy. It was impressive and it was difficult before, and now it's easy, and we're going to begin to automate it. So in summary, when we're thinking about these data commons, relative to a lot of these data sets, 500,000, 500,000, hundreds of thousands, the NIH data is relatively small and it's messy, it's heterogeneous, right? It's a lot of different sources, a lot of different people with a lot of different interests trying to put it together. I think one of the strengths of the HGSC and one of the things we've capitalized on in our partnership is taking that heterogeneity, figuring out how to harmonize it and turning that into best practices. We really know what we're doing with these data. And efficiency is good. We've done a ton of things efficient, but precision is better, and it's critical. If you have messy data, you gotta make sure it's clean. So again, this is the Sentient thing. Yes, they're really fast, but that's not as important as the fact that they got it right, and they got it right perfectly. So the bottom line is that if you do data commons right, it drives innovation. Again, you make your difficult tasks routine, and you make your routine tasks automatic. And that's exactly what happened with charge. That's what's happening with structural variation. With eMERGE, that's what we're automating with Neptune. And I think that's sort of one of the differences that, working in academia with these messy data differs from some of these giant machines that crank things out. So there's a nice balance and give and take between developing the best practices and pushing them forward. I think I filled 5 minutes on 2 slides. So I will go to my acknowledgements. My team's the Next-Gen Sequencing Informatics, which I did inherit and drastically improve on from Jeff. And this is the team. UT Health, this is Eric Borwinkle's team. Again, everyone at DNAnexus, and I'm going to mention Maria and Geet. We've been working on getting our local workflow, HGV, pointed at the cloud. It's going to be fantastic. It's just not ready for to go into in this venue right now, but by next year, that will be what the talk is about if you guys invite me back. Sentient guys, I love you. This is our leadership. This is basically the group of us. We go and eat Mexican every 3 weeks and decide what's going on in the world of genomics. And then other leaders here at the HGSC who are critical to putting out quality data. So with that, I am glad to answer questions from anyone except David.
There you go. All right.
I'm kidding. Go for it, man. I know you can't help yourself.
It's all true. So actually, while that slide is up, first of all, fantastic talk. I have a sneaking suspicion you will be back. The DNAnexus folks who were there are a few people you specifically gave a shout out to who, if they're, when we see them, I'd love them to stand up. Andrew Carroll, all the people on that slide who are here, Mike, Lynn, Martin, Maria, Geet. You guys should really see the people who,
Yes. Please clap for them for me. I don't have the budget to afford such quality people, so I'm glad to have you guys helping.
It's an exceptional science team, and you know, we work really well with inspirational partners. All right, one question, and then let's get on with it. We have sort of quicker talks coming up. One question for Will. Anyone? All right, well, he's around, and you can't miss him, so make sure you catch him. Thank you very much.
Thanks.